This video explains how to perform transmission electron microscopy, also known as TEM, on the Drosophila embryonic heart. The heart has emerged as a valuable model system for studying cell migration, cell-cell adhesion, and cell shape changes. This technique allows visualization of heart lumen shape, as well as ultrastructural components such as cell membranes, cell-cell junctions, and basement membrane. To begin this procedure, a fixation solution appropriate for transmission electron microscopy of Drosophila embryos is prepared. Next, a 0 to 20 hour collection of decorianated embryos are placed into the fixation solution. When finished, embryos are removed from the fixation solution and hand-devitrinized while attached to double-sided tape in a petri dish containing buffer. Devitrinized embryos float in the buffer and are recovered with a pipette. Then, embryos are post-fixed and undergo a dehydration gradient. Once infiltrated with resin, they are transferred to an embedding mold where they are aligned. The resulting block is cut with a diamond knife. Sections are picked up on copper grids, stained, and examined.
Hi, I'm Nadine Soplop from the laboratory of Sunita Kramer in the Department of Pathology and Laboratory Medicine at the University of Medicine and Dentistry of New Jersey at Robert Wood Johnson Medical School.
I'm Raj Patel. I'm the electron microscopist, also from the Department of Pathology. Today, we will show you a procedure for preparing and imaging Drosophila embryos for transmission electron microscopy.
We use this procedure in our laboratory to study Drosophila heart morphogenesis. So, let's get started!
Before handling the Drosophila embryos, freshly